Are cancer cells all the same, or do they vary from patient to patient? Are there differences in the cells within a cancer tumor? How could these differences affect treatment options? And why might differences in transcription exist in different cells within the tumor? Welcome back to NeuroSciQ for another episode. In this week's episode, we're going to be talking about tumor heterogeneity. So sit tight and stay tuned. Despite what many of us believe, tumors are not just a mass of cells. Tumors are actually quite complex, and the cancer cells that comprise them vary not only between patients, but also within patients. Obviously, with this added layer of complexity, we have obstacles to our treatment options. So today we're going to talk about inter-tumor heterogeneity and intra-tumor heterogeneity. So first we'll look at inter as a prefix. Inter with an E is when we have variation between patients. So inter, the way I would always remember it is it has the E in it, and between has the E in it as well. So differences between patients has long been recognized, and it's something that people tend to grasp a little more easily. For instance, we know that we're all different, and so it would make sense that our cancer tumors would be different as well. Now, this happens because of patient-specific factors. So for instance, there might be germline mutations, there might be different somatic mutation profiles, but also the environment that a person lives in can influence their tumor as well. So for glioblastoma, we have IDH mutant, we have the IDH-wildtype, and we also have some histone mutations that occur between patients. The importance of these mutations in this intertumor heterogeneity is that they affect the treatment outcome. For instance, IDH mutations tend to be more indolent, and so they're less aggressive tumors, whereas tumors with histone mutations tend to be more aggressive in comparison to IDH mutants. If you want to know a little more about that, Dr. Andrew Gao gave a lecture with us, and he touched on those different types of tumors in that lecture. Now, the next prefix, intra is when we look within a patient. So A for the same, and intratumor heterogeneity is when we have differences in the different regions within a tumor. So we can have spatial heterogeneity, where we have genetic diverse tumor subpopulations based on the location, and this can happen because different locations of a tumor will be exposed to different environmental factors, and so this can select for different types of mutations within the cancer cells. Over time, we also have temporal heterogeneity, and so this means that we can have differences in the tumor as it changes and as it adapts in the human body. So in this image, we see the spatial heterogeneity, where we have a tumor within the person. We see different types of tumors in different locations, but also the tumor has different cells in it as well. Temporal heterogeneity are the changes that happen over time, so we see that the tumor starts off with different cells, and as time progresses, the cells change and mutate further. Now, this temporal heterogeneity that we see might not only be due to these natural reasons, but it can also be due to the way that the tumor responds to treatment. So you see line 1, line 2, and line 3. Those could be changes in the tumor after different treatment options. So, in case you didn't know, heterogeneity is the quality or state of being diverse. And so it is the opposite of homogeneity, which is where everything is the same. Why is heterogeneity so important? Well, intratumor heterogeneity seems to fuel resistance. We have these cells that are phenotypically and genetically different with distinct molecular signatures, and so they are going to perform differently against different treatments. So for instance, if we have chemotherapy, certain cells might be affected by the chemotherapy, but other cells could be resistant. And so heterogeneity is important in this way because we need to be able to target all the cells without having a couple of resistant cells escape and make it through. So cancer starts off with mutations. We start off with these non-malignant cells that progress into malignant cells based off their alterations in their cell profile. So these alterations tend to promote cellular proliferations, and that would be things like evasion of growth suppression and apoptotic signals. So growth suppression factors prevent the growth, and apoptotic signals, apoptosis is cellular death. So without apoptotic signals to shut down cells that are detrimental or that have reached the end of their cell cycle, we have continued growth, and then that is how you develop a tumor, where you have all these cells that aren't doing anything good for the body. We also have signals that cause angiogenesis, and we've touched on this in the last couple of videos. So cancer cells tend to promote angiogenesis, which is the growth of new vessels to supply blood to the area. And we also see the activation of metastasis and tissue invasion. Now these mutations are random, and even after the transformation, cancer continues to change. We have genetic, transcriptiomic, epigenetic, and phenotypic factors that influence. So the genetic factors are just the gene profiles. That would be things like a single base mutation or perhaps a whole chromosome deletion. Transcriptomic comes down to the RNA level. And then we have the epigenetic level, which is alterations to the mRNA, and we also have phenotypic variations, obviously in the end, based on the protein expressed. Now there's a quote from this paper that says, "Intratumoral heterogeneity drives the evolution of cancers and fosters drug resistance." But why does this heterogeneity exist? Well, with cancer we have genomic instability. There's mutations because of mutagens, internal error where the DNA replication perhaps just messes up and it goes uncorrected. And then there's also the elevated mutation rate theory. So in some cancers we have chromosome losses, and so the theory is that because of this loss in a chromosome, there's instability because of chromosome level changes. So we have gain or loss of genome segments, and so the replication of DNA could be messed up because of this. That could lead to a higher rate of mutation. Because we have such a high rate of mutation happening in cancer cells, it's expected that they're going to be different from each other, and so we have heterogeneity. There's also the selection hypothesis. Now this was a hypothesis that was first presented by Peter Nowell in 1976, and it's kind of similar to Darwin's natural selection theory. So we know that tumor initiation is random, and that we have an initial mutation that leads to a growth advantage, and that causes neoplastic proliferation. But what we haven't looked into as much is the fact that evolutionary pressures will actually select for different mutations. So we have this high rate of mutation, and different areas of the body have different environments. So there will be different cells that are selected for and that fare better than the others. This is how subclonal tumor cell populations emerge. So we see that in this image here, where we start off with non-malignant tissue, and then there's a mutation that creates a cancer cell with a growth advantage, and because of the genetic instability, new cells arise. Now these new cells might fare better in the environment, and so they increase their amount of subclones. How do divergent populations emerge? Well, that's just the accumulation of mutations over time. So depending on the mutations, we can have different clonal populations, and depending on the location in the tumor, certain cells might survive, and so we get this heterogeneous mass. As you can see with all the different cell types, would be harder to target versus a mass of a single cell type. So what can happen is if we have a drug-resistant cell, then after treatment, that one resistant cell will likely progress into another mass of cells. Now, since we're a neuroscience channel, it's nice to talk about heterogeneity and all, but how does this link back to the brain? Now, if we look at glioblastomas, which are brain tumors, then this has an obvious link. Neftel and colleagues published a paper back in 2019 in August, so just under a year ago, and their paper looked at GBM, and what they did was they looked at single cell analysis. They also looked at experimental models to discern the heterogeneity within a GBM. And they revealed through single cell analysis of the GBMs that there are multiple cellular states. What they found were specifically 4 genetic states that drive heterogeneity. And these states were oligodendrocyte progenitor cell-like, neural progenitor-like, astrocytic-like, and mesenchymal-like. Lineage tracing shows that these states can transform into one another, and they also found the environment influences the cellular state. In particular, they related these different cellular states to copy number amplifications of CDK4, EGFR, PDGFRA, and NF1 locus mutations. Now, the EGFR mutations were specifically related to the Cancer Genome Atlas Classical subgroup, and PDGFRA were specifically related to proneural subgroups. All 4 states may exist in tumors with mutations in one of those 4 events. It's just that depending on the mutation, the states will be favored in different proportions. Now that brings us to what exactly are these Cancer Genome Atlas subgroups. We have classical, proneural, mesenchymal, and mixed. These subgroups would be examples of intertumor heterogeneity. What the Cancer Genome Atlas does is groups tumors based off of mutations that they have and cellular profiling into one of these groups. Back to what was done in this paper published in Cell. Now what they did was single-cell RNA sequencing of 28 tumors. Starting off, the first figure in the paper looked at the single-cell RNA profiling of these tumors, and what we see in figure 1C is that these tumor cells tended to cluster together based on the tumor they were from. So we see the different colors which represent different tumors, and the cells from the same tumor tend to cluster together, which is something we would expect seeing as they're coming from the same place, so they probably share similar profiling if they all came from the same progenitor cell. What they did with the single-cell RNA sequencing afterwards was hierarchical clustering. And we see in the next figure that these clustered into what seemed to be 4 groups. Now those groups did split up a little bit further down the road, but what they decided was that they would keep these 4 bulk groups of mesenchymal-like, astrocytic-like, and neural progenitor cell-like, as well as oligodendrocyte progenitor-like. The mesenchymal group also had 2 subgroups, mes-like 1 and mes-like 2, and the NPC-like also had 2 subgroups as well. So that's where they got a total of 6 groups from, but they decided to stick to the 4 based off of the bulk separation. Now, these 4 metamodules that they chose varied based on copy number aberrations and mutations. What they saw after was that cells do tend to fall in one of these signature groups. So after assigning the signature and mapping out the cells from different tumors, they found that the cells would fall into either OPC-like, NPC-like, mesenchymal-like, or astrocytic-like. So these cells fall into their specific 4 groups, and what the chart also shows is various differences based on the location of the cell in the cell cycle. So these are cycling cells, which means they are going through replication, and red would be higher density, and when a cell is more dense, it tends to be actively going through mitosis. So the mitotic cells seem to vary from the subtypes as they cluster more in the middle, and they would be more similar to one another as they're going through mitosis, but the ones that are not actively undergoing mitosis but are still cycling would seem to separate into their own groups more. So the bottom line is that we are seeing this intra-tumor heterogeneity. What we look at later is how the different types in the Cancer Genome Atlas vary based on these 4 metamodules. So the Cancer Genome Atlas already has its defined groups of GBM, which we mentioned earlier. What we see when we look at them is that the proneural group, the mesenchymal group, the classical group, and the mixed group vary in cell population. So the 4 metamodules that were identified by Neftel et al. Would occur in different amounts based on the subtype. So we see that the proneural group seems to have more NPC-like cells, or in some cases more OPC-like cells. The mesenchymal group has more mesenchymal-like cells. The classical group seems to be more astrocytic, and the mixed group seems to have more of an even separation of cell types. Now each pie chart is a different tumor in this figure, and then the different colors show the different classification of cells and their abundance in these tumors. So the Cancer Genome Atlas does bulk analysis, and what this paper did was when it compared the bulk analysis of the Cancer Genome Atlas to their single-cell RNA analysis, they saw that, well, for example, a tumor with mostly mesenchymal cells would be grouped as a mesenchymal tumor based on the Cancer Genome Atlas. It does have other cell types in it as well. And so we see this in all the other tumor types as well, where we have the bulk of the tumor giving us a certain reading, and the single-cell RNA analysis would reveal that all 4 cell types are there as well. This is something that has important implications for treatment, as we can't just target the one cell type based on bulk analysis, because those other cell types do exist in the tumor still. The next thing that was done in this experiment was to confirm whether or not these cell types come from one another, or whether or not they can transform to one another. And the way that this was done was by using a lentivirus to insert the barcode. Now let's look at the one half of this figure first. So starting with figure A, what we see here is that they used single cell sorting. They collected groups of cells, and they took these cells and created a xenograft for the mice. So basically they're inserting a tumor in the mouse. What they saw was that these tumors, no matter what kind of distribution of cells they put in the mouse brain, the tumor tended to return to its original distribution. So we have what looks like an astrocytic tumor. So you see in the first panel that all the tumors kind of resemble the same group with more astrocytic-like cells. In the second panel, it seems like they injected more NPC-like cells, but what we see afterwards is that the tumor actually progressed back to more of an astrocytic-like distribution. Again, they did it with more mesenchymal cells, and again, the tumor progressed back to more of its original state. What they also did was they inserted a lentivirus for just one barcode of one of the metamodules. And what they saw was that even with just the one barcode, the mice, in the end, ended up having all 4 of the cell types in their final tumor. Now this is very important because for treatment we have this issue that if one of the subtypes is resistant to the treatment and it makes it out, then what can happen is we can just have proliferation and recreation of all the other cell groups. This adds an extra barrier to cancer treatment. And so it's something that scientists have started considering and looking at cocktails of drugs to treat a tumor rather than just one drug, which might be targeting only a subset of the tumor cells. It's just something we need to think about in the future as we look for treatments for these cancers. Not only for GBM, but also for all other types of cancers as well. Thank you for watching the video today. If you have any questions, leave them down below. Make sure you like, comment, and subscribe. And also, if you have suggestions for future videos, feel free to leave them down below as well. That's all. See you next week.